We're doing research on the soil biodiversity in agricultural systems in South Africa, and we especially work on a group called Springtails or Collembola. They are extremely abundant in the soil, but they're very small, so people don't know much about them. They're about 2 millimeters in size. What makes them very different from insects is that they have an appendage under them called a furca, which is like a springing organ, which they use to escape predators. And they also have a ventral tube, which they use for gas exchange. Springtails also don't have wings, and they actually evolved as early as 400 million years ago. So they evolved way before any insects. What makes them very unique as well in terms of soil studies is they don't have a hard exoskeleton like spiders or beetles or other insects, so they actually breathe through their skin, which means they're in direct contact with the soil. So they're very sensitive to changes. So our project is to look at, are they good indicators in agricultural systems for soil health? It's usually thought that you look at soil chemical components, but actually the soil biodiversity and indicators in the soil, live organisms can be great indicators of soil health. For example, nematodes are often used, but in South Africa, springtails have not been used before as bioindicators. So we're looking at what kind of, how many species there are and how many there are. In other studies, they've shown there can be up to 40,000 individual springtails per square meter. So that is a lot of individuals. So, which is also a reason why people have not really worked on them. It takes a lot of time to process samples, to identify them. But we're finally at a point, and we have international collaborators and South African collaborators, that we're at a point now where we can actually identify them. We use taxonomic experts, but we also use molecular methods to identify the species. So we're looking at springtails. We're looking at what I said, they're very sensitive to soil moisture because of their that they're direct in contact with the soil. So especially in conservation agricultural systems, we're finding now is that they are extremely abundant, and it's probably likely due to the permanent cover crops and the crops on the soil. And another feature of them, they are extremely important as prey for other arthropods like spiders, pseudoscorpions, and mites. So the research has shown that these arthropods, these predators, feed on these springtails in months where pest species such as aphids are actually low in abundance. These spiders or all these predators can feed on springtails, and when the season comes, when the pest comes back in abundance, these natural predators are actually in high abundance to actually attack these pest species. So they're very, very important as prey for these predators. I want to tell you a bit about other important soil fauna we get that are often overlooked. I think most people know about nematodes, not always in a good way, but there's actually very beneficial nematodes. There's even smaller things than springtails called tardigrades, and they're very abundant. They're actually within the pores and in between all the soil particles. You get tardigrades, you get all kinds of mites. So you get two large groups of mites. There's either predatory mites, so again, they are very important to control pest species. And you also get mites that feed also on fungus, so they're actually also important in the decomposition process. Then you get things like that people have never heard of, like proturans and diplurans. They're also wingless arthropods in the same big group as springtails, and they're also important in decomposition, but there's very few people studying them, especially in South Africa, so we don't know much about them. Other groups that we also don't know much about, so we do know a lot about ants, termites, beetles, the bigger things which you can see. So we're trying to look at things that you actually can't see. You need a microscope to study these animals. And of course, you get all kinds of decomposers like millipedes, and everyone is familiar with earthworms. So springtails do similar work than earthworms, but of course they're a fraction of the size. That is why they're very abundant, and they eat, springtails eat fungus, they eat fungal hyphae, fungal spores, and then they also eat dead organic material. So they're very important in decomposition and to deliver nutrients to the plants. And then the largest, of course, animals in the soil you get are vertebrates like skinks or even moles. So in our review, we sort of looked at all arthropods in all soil biota, from the microorganisms, bacteria, fungi, all the way through to larger insects and vertebrates. People are often not aware of the very complicated soil food web that you're walking over every day. You get all these, you get basically litter. For example, especially in conservation agriculture, you get a huge amount of leaf litter. And that really promotes soil biodiversity by creating this microhabitat for all these amazing animals in your soil. So there you have your mites, you have all the sort of the prey for all the predators. So springtails, mites, all these things. And they in turn are eaten, of course, by the predators, as I mentioned. But also these grazers graze on the fungi, which then creates an increase in decomposition. So you have a system, if there's a lot of leaf litter and a lot of cover, you create a system with more fungus, which promotes the decomposition and then the nutrient recycling. So it's not just one quick fix, it's not just one invertebrate you need. You actually need this whole complicated food web to work to make a really healthy system. And the importance of that is then you have really resilient soil, which means you can get through circumstances that you won't necessarily get, or when the soil is very dead, that reduces the soil biodiversity. So you might only have one very resistant springtail, for example, but the spiders might be gone. So you have a very sort of unbalanced system if there's not this amazing microhabitat created for them. In a conventional system, you usually have less biodiversity, as I mentioned, but then you have a system dominated by bacteria, which is not always beneficial, especially for plant recycling. So these interactions between all these soil organisms and the increase in soil biodiversity is really what makes soil resilient and is an adaptable, flexible system, especially with increasing in climate change and extreme events. These systems can really be resilient. So you do need as high soil biodiversity as possible. So this is just a sample I want to show you that we collected from this trial. This is actually from the soil and a bit of the leaf litter. And as you can see, the springtails really dominate these samples, which is a good sign. They are mostly from one species in this sample. You can see here's their body, and in comparison to the soil particles, you can see their size. These are their legs. They have 6 legs, and the furca. This is from the family Hypogastruridae, and extremely abundant. So in this sample, there's probably about more than 500 specimens. Again, as I mentioned, this is the reason why many people don't work on this group. It's very long processing time. Here you can see in the sample there's an ant, and you can see how large the ant is in comparison to the springtails. We also have quite a few other insects. This is a type of a midge or a dipteran. And then we have a lot of mites and all kinds of different things in here. So very diverse arthropod mix here. This is also a ferocious looking mite there. So this is a springtail we collected also on this farm, and this really shows you nicely the morphology of springtails. So they have, they also have 6 legs, they're wingless, and this is what's called the furca. Usually it's tucked in underneath their bodies, and it's got a little clasp, so it's usually tucked in under their bodies, and then when a predator comes or when they are startled, they actually release it by pressure, and then they jump away. There's often a saying that says that they can jump as high as a human jumping over the Eiffel Tower. And the things we use to identify them are usually their body shape, so how many thoracic segments they have visible, how many abdominal segments, the number of their eyes, the length of the antenna. So to identify springtails are quite tricky. Like I mentioned, we work with taxonomists, and that look at the types of their hair, they look at all kinds of things, but also we use molecular methods, which is called DNA barcoding, which looks at a gene that can tell us at least that they will say these 2 species are the same or not, and they can also say if a species is introduced. Usually that's a good indicator of, in natural systems, if a species is invasive or introduced, it's usually a disturbed habitat, and when there's indigenous species, it's usually not as disturbed in a pristine habitat, which is what we found in the fynbos in the Western Cape. So these specimens that we collect here on the farm will also be what we call barcoded. So they will be barcoded, and then we will know if they're indigenous or not. And then we do actually expect that we can find new species here because they're not very well sampled in South Africa, although we have more and more people working on them now. And globally, there is a growing number of people working on them. In the world, there's about 8,500 to 9,000 species described. In South Africa, from the literature, we know of only about 130 species, but we expect about 1,000 species to occur in South Africa from our collections. Yes, so it's a very exciting group to work on, and they are just so cute, and we actually walk over them all the time and we don't know it. They've found from the Arctic to the Antarctic, they found from the sea sand, they're in sea sand, to the highest mountain peaks in the world, in the Himalayas. So they actually occur everywhere in huge abundances. There's even a few species in deserts. But due to their very sensitivity to soil moisture, because they breathe through their skin, so they're very prone to desiccation. They're not as abundant, of course, in deserts, but they're very, very abundant in humid tropical areas. So here we have a sample we collected from the field here. And as you can see, it's got really rich cover material. So as I said, that microhabitat for all the soil fauna. And the way we get them out is to put them on what's called a Talgren or a Belize funnel. It's just a funnel with some mesh on, and then underneath there's a little vial. Let me just do this like this. So just over time, all the animals in the leaf litter and soil move down and fall down into the vial. And then we collect them either in ethanol, but if we want to study them alive, which we can do, we just collect them like this. And I'll show you now all the activity that's in here. So everything falls down here, from the spiders, the mites, and then the springtails, which is the dominant group, at least on this farm. An easy way to quickly check if you've got what kind of life you have in your garden or in the field or wherever is to take some of that soil or litter sort of cover material, put it in a kitchen sieve like I did and a bowl, and just shake it like this, and then you'll see when you remove it, you'll see all the life that's in your field. So here I can see a little indigenous fish moth. There's lots of springtails running around here, and a little spider. Of course, the cleaner, if there's not enough soil, you can see more, but this is an easy way to see how many groups of soil fauna you've got. There's a little springtail. And I usually collect them like this. We're using an aspirator, which is a little entomological contraption like this. So there's mesh on one end, and there's a tube, and then you suck the end with the mesh just to collect them. And I'll show you under the microscope how they look. So from that sample, we were just extracting. We could see all the life in there. Lots of springtails. There was a mite running around. There's just a whole life in the soil that one never thinks about. And they just go on and eat and do their thing. In some very, very poor soil areas, springtails actually contribute to making soil by basically with their fecal pellets. And also by moving through the soil, they aerate the soil. Just want to see if I can find some more. They're trying to get away from the light, so they're always hiding. Usually if you find springtails that are very white, it's an indication that they adapted to living deep in the soil, while ones like this that are a bit darker, they're more adapted to living on top of soil. And then you get ones with those very long furcas, which I showed you, and they are adapted to, they can actually move up and down plants as well. So springtails are not just in the soil and in leaf litter, they can actually live on plants as well, where they eat fungus and bacteria. There's a little guy. He's moving so fast. Because, as you can see, it's very easy to collect springtails. They're very small. They're very easy to actually keep alive. That's why they make very good model organisms or bioindicators, and we can keep them alive in the laboratory. We can breed them and do all kinds of experiments on them. So they have been used for decades as ecotoxicology indicators, so to see how polluted soil, sort of different types of soils are. And more people are looking at their physiology, so really what is the hottest temperatures they can handle, what are the coldest temperatures they can handle, and actually use this to see what will the impact be of climate change on soil life. This is not just on springtails, but on all kinds of soil biota. So for example, we did a study where we looked at the thermal tolerances of springtails, and we found that introduced species, so that is species that are from other countries accidentally introduced, actually have much higher tolerances to higher temperatures than indigenous species. And with climate change, and even soils will warm up a bit, we actually found that these invasive species will probably are predicted to dominate the soils. What this means in terms of function, we are not sure yet. There's experiments on the way to look at that. But we do know that invasive species generally reduce biodiversity, so reduce the number of species in soil, which is not necessarily a good thing. So that is very interesting research that one can do on springtails. We also found that the invasive species are more prone to be more resistant to desiccation. So the future projections in most places are that soil will get warmer and drier, which means that these introduced species or very aggressive invaders might just tend to dominate systems. So the more diverse your soil is in terms of not only the number of species, but the number of plants, that your soils can be much more resistant to these climate change effects, these biological effects which aren't always considered.